Okay, so we're going to get into chapter 16 with this lecture. Going into the senses. This one, uh, the general senses that we start with kind of the general sense functions and some of the general senses. They're not so intense, but then we're really going to get into vision, hearing, and equilibrium, and that will take up the bulk of the second lecture, the second part of this lecture. So starting with this, so sensory receptors, they provide information about the external and internal environments, external and internal. Remember that we have our visceral receptors providing information about our organs. They respond to a stimulus. Each type of receptor responds best to a type of stimulus. So light receptors for the eye, sound energy for ear receptors, there's a pairing. There's a type of receptor for a type of stimulus. Transducers are going to convert that stimulus energy into electrical energy. Remember, with all of these kind of sensory inputs, we have vibrations, we have mechanoreceptors detecting touch and pressure, we have light receptors receiving light. Those all have to be converted into some type of electrical signal that can then be interpreted by our brain. So we're going from the stimulus being detected by receptors, converting that to some type of electrical signal. Those electrical signals are then turned into action potentials that are conveyed to the CNS for interpretation. So the receptors have a receptive field, which is the distribution area of the endings of a sensory neuron. So that's kind of saying that that's the area that receptor is covering, is going to be detecting stimuli from. So the smaller receptive field allows for more precise stimulus localization. In our fingertips of our hands, we have very small receptive fields, so more neurons, smaller receptive fields for each of those neurons that allow for more discrete location of touch. Whereas kind of here on the back of your shoulder, you have a much larger receptive field. So you have maybe fewer neurons that are detecting across a larger amount of space. The sensation, so a stimulus, that we are consciously aware of is called a sensation, right? So you might have the sensation of being cold, the sensation of hot in your mouth if you're tasting something, sensation of touch. Those are all consciously perceived stimuli. To enter consciousness, signals must reach the cerebral cortex, so that outer portion of our cerebrum, and only a fraction of stimuli result in sensations. That's kind of lucky. If we were actually consciously aware of every bit of stimuli information, that would be overwhelming for us. A lot of sensory input goes to other areas of the brain. So remember, there are just those different portions of the brain that are specialized to interpret specific information. So blood pressure signals go into the brain stem and the medulla and the pons to help regulate our cardiovascular responses. And receptors provide CNS information about stimulus modality, location, intensity, and duration. So those are the four things that are going to be conveyed to our central nervous system. Modality, so the type of stimulus that it is, where it is, how intense it is, and is it prolonged or is it quick? So modality is the type of stimulus based on labeled line. The brain interprets optic nerve signals to occipital lobe as visual, cochlear nerve signals to the temporal lobe as auditory. This is just saying that our nervous system has a way of being able to direct sensory information to the appropriate area of the brain. The location of each stimulus is determined by by which receptive field is active. So those receptive fields all throughout our body then correlate to a spot in the postcentral gyrus in our brain. Remember, there's that central gyrus, and then you have the postcentral, which is housing your sensory information, and the prefrontal, which is precentral, which is regulating your motor output. Okay, so this intensity of stimulus is determined by the frequency of nerve signals to the CNS. Remember, we said that if in the nervous system, if it wants to increase the intensity of a signal, it increases the frequency of that signal. The stronger stimuli causes more neurons to fire and cause sensitive neurons to fire more frequently. So more intense stimulus, the hotter something is, the sharper something is, the rougher clothing might be for you. Something like burlap that's very rough. That's going to send even more stimuli to say, hey, this is a very, very intense, or even more frequency of signals to signify an intense stimulus. Adaptation, so receptor adaptation. This kind of goes along with the fact that we don't detect every bit of every stimulus that we can be perceived. Receptor adaptation helps to determine duration. So, this I always kind of go immediately to clothes. So, after a while you stop, you put your shirt on, you put your clothes on, but then after a while you stop realizing that you're wearing them. Your skin receptors become adapted to that stimulus. So adaptation is decreased sensitivity to continuous stimuli. Tonic receptors are limited adaptation. They continuously are going to respond. Head position receptors in the inner ear, all pain receptors, so your nociceptors are going to all be tonic receptors where they don't really adapt to that stimulus. They're going to keep signaling it. Phasic receptors adapt rapidly and only respond to new stimuli. These are going to be your pressure receptors. So again, kind of around the receptors on your hair follicles, in your hands, as you're holding something, it's that thing where you're holding your phone. It's like, where is my phone? I have no idea where it is. And you don't even realize that you're holding it in your hand because it's just something that we've become adapted to. Let me pull myself down just a little bit. There we go. Okay. Receptor distribution. So there are general sense receptors and special sense receptors. The general sense receptors are going to be simple structures distributed all throughout the body. You have somatic and visceral sensory receptors. The somatic ones, again, going to tactile receptors, proprioceptors. Visceral sensory receptors going to be chemoreceptors, baroreceptors in your organs, things like that. Your special sense receptors are going to be for olfaction, gustation, vision, audition, and equilibrium. Audition is not a special sense for making you great on American Idol. It is for hearing. All right, stimulus origin. There are exteroceptors, interoceptors, and proprioceptors. Exteroceptors are going to detect stimuli from the external environment. You can think about your eyes, your skin, and your mucous membranes. Your interoceptors detect stimuli from your internal organs, your blood vessels, your GI tract. And your proprioceptors detect body and limb movements. So your proprioception is your ability to detect your orientation in space. There is a modality of stimulus. So there are 5 different types of receptor modalities. You have chemoreceptors. You should know all of these. There are chemoreceptors that are detecting the chemicals dissolved in your fluid, in your body fluids. There are thermoreceptors that are detecting changes in temperature. You have photoreceptors detecting change for light intensity, color, and movement. Mechanoreceptors detecting distortions of cell membranes, so pressure receptors, vibration, stretch, all those things. And nociceptors detecting your painful stimuli, somatic and visceral nociceptors. All right, tactile receptors. So this is a specific type of one. These are abundant mechanoreceptors of the skin and mucous membranes. The endings can be encapsulated or unencapsulated. We looked at these when we did the integumentary system. If you remember, you have your epidermis, your dermis, and then your subcutaneous layer. There we go. All right, starting with the unencapsulated tactile receptors. The dendritic endings of sensory neurons with no protective covering. They have no connective tissue covering wrapped around them, so they're just exposed. You have most basically your free nerve endings, which are terminal ends of sensory neuron dendrites. They're the simplest tactile receptors. They are close to the skin surface and your mucous membranes, mainly for pain and temperature, but also light touch and light pressure. Maybe phasic or tonic, so they can either adapt quickly or slowly. It just depends. You have your root hair plexuses. They wrap around hair follicles. They're located deeper in the layer of the dermis. They detect hair displacement, and these are phasic receptors. So these are times what are detecting our stuff up against your skin as the hair gets distorted. You have these root hair plexuses that are detecting that. They're phasic receptors, so they're going to adapt quickly. You also have tactile discs, which are flattened endings of sensory neurons extending to tactile cells. Tactile cells are specialized epithelial cells in the basal layer of the epidermis, so that kind of bottom layer of the epidermis. They respond to light touch, and these are tonic receptors, so they're not going to really adapt. Here is just a depiction of those. You can see the free nerve endings, these wrapped around the root hairs, and then these tactile discs up against tactile cells, transmitting that pressure signal. All right, now looking at the encapsulated tactile receptors, these are going to have some type of connective tissue wrapping covering around them. You have end bulbs that are ensheathed in that connective tissue located in your dermis and mucous membranes. They detect pressure and low frequency vibration and are tonic receptors. Your lamellated corpuscles are wrapped in the neurolemnocytes and concentric layers of connective tissue, so kind of almost similar to myelin, I guess. They're located deeper in the dermis, the hypodermis, and some organ walls. Remember, the hypodermis is just another name for the subcutaneous layer. They detect deep pressure, coarse touch, and high frequency vibrations and are phasic receptors. Your bulbous corpuscles are within the dermis and subcutaneous layers. They detect deep pressure and skin distortion. And again, if they're in the subcutaneous layer, they're not going to be able to detect light touch. They're going to have to detect deeper pressure. And these are tonic. Your tactile corpuscles, intertwined endings wrapped in modified neurolemnocytes. They're in the dermal papillae. So if you remember, the dermal papillae are what contain some of those vessels to nourish the epidermis. Discriminative light touch allow recognition of texture and shape, and they're phasic. So again, if they're in the dermal papillae, that's fairly superficial in the dermal layer, so they can still be able to detect light touch, and they're phasic. Referred pain. This is actually very interesting, something that as a massage therapist I deal with a lot and have several stories about. So referred pain is an accurate localization of sensory signals. You have signals from viscera, so from your organs, perceived as originating from your skin or your muscles. You have many somatic and visceral sensory neurons and signals via the same ascending tracts within the spinal cord, so those signals can then get confused. The somatosensory cortex is unable to determine the true source. So essentially what's happening is that you have all of these sensory neurons coming from your somatic body, your visceral body, or your somatic receptors and visceral receptors. And as they come up the same ascending sensory tracts in your spinal cord, it's hard to really accurately determine always where those signals are coming from. One rather sad example I have of this is a friend who for months was having this really bad low back pain. She saw massage therapists, chiropractors, physical therapists. It was always right in her vertebrae. She couldn't quite figure it out. And it turns out that it was stage 4 pancreatic cancer. It took, going to the doctor and really getting a full fledged out diagnosis and workup to determine that. It's, one of those things that it's just how the organ refers its sensory pain. So this is a very interesting field. There is that clinical relevance to it beyond just that example that I gave. You also have heart attack symptoms. Pain in the heart in the myocardium can refer to different portions of the somatic body, to the pectoral region, the medial arm. You have kidney and ureter pain referring to the inferior abdomen. It's also often conveyed along sympathetic nerves, but occasionally on parasympathetic nerves. So your bladder pain can be conveyed through your sacral parasympathetic nerves and refer to your buttocks. So it's all over, lots of different nerve signals getting crisscrossed and wrapped around. Luckily, these tend to be fairly predictable, not always, but sometimes. And so we can start to pinpoint, okay, if you're having this pain in certain referred areas that are common, we can know where to look instead. Here are some of those just kind of being mapped out. So heart referred pain, liver and gallbladder referred pain, your stomach, your pancreas, all that kind of stuff. If this seems or feels a little too woo-woo, some of you it feels a little bit like we're going into acupuncture and acupressure, it's okay. It still all works. Referred pain is a real thing. All right, now getting to some of our specific senses. Olfaction, the sense of smell. The olfaction is detection of odorants dissolved in the air. They are volatile materials. Odorants are volatile molecules, dissolved in the nasal mucus and detected by chemoreceptors. So if it's going to be a molecule, it's going to be detected by a chemoreceptor. And notice this, it has to be dissolved in that nasal mucus. When we don't have as much of nasal mucus, those odorants don't have a place to dissolve. They need that kind of fluid substance, that medium to be able to dissolve into to interact appropriately with the chemoreceptors. These provide information about food, people, and danger. And we can distinguish thousands of different odors. Just the way that the molecules combine sends a combination of specific nerve signals that our brain can then interpret into actual information. Your olfactory epithelium is your sensory receptor organ. And notice this, it says that it's an organ, which means that it has multiple tissue types. Okay, that's just kind of thinking about how we put things together. So your olfactory epithelium is your sensory receptor organ. It's in the superior region of your nasal cavity, so the upper region of your nasal cavity, and has 3 cell types. There are olfactory receptor cells that are detecting odors, so olfactory receptor cells, that should not be, that name tells you what it does. You have supporting cells, which sustain the receptors. Again, it tells you what it does. And basal cells that replace olfactory receptor cells every 40 to 60 days. So these are replaced every 2.5 to 3 months, or I'm sorry, 1.5 to 2 months. You're going to have your basal cells, these stem cells, replacing your olfactory receptor cells. Rejuvenation and sensitivity of receptors decline with aging. If you think about it, mucous membranes tend to be exposed. They are exposed to the external environment, so they do undergo damage and irritation faster than some of our internal serous membranes. So the olfactory membrane is replaced again. It's said every 40 to 60 days. But as we age, that starts to slow down. Your sense of smell starts to decline, and it's impaired. You have a lamina propria as well as the epithelium. It has areolar connective tissue, which is the internal layer to the olfactory epithelium. It's connective tissue, so it's going to be housing blood vessels, nerves, and olfactory glands. That's what connective tissue does. Your olfactory glands help to form mucus covering the olfactory epithelium, so that mucus that we need to dissolve odorants. And here is just a nice little image of that. As we get into this, we're about to get into some of the more nervous structures that we have of it. But you can see this mucus layer. This is what's going to be internal to your, or continuous with your nasal cavity. You have the olfactory epithelium right here with different receptor cell axons, these dendrites. You can see these olfactory hairs extending into this mucus to be able to detect odor molecules. There are these basal cells, supporting cells. Here's that lamina propria layer, attaching it to the cribriform plate of the skull, and then we have the olfactory bulb, which is part of that olfactory nerve. Okay, so olfactory receptor cells are the primary neurons in the sensory pathway for smell. They have a bipolar structure, which means they're going to have that single dendrite and an unmyelinated axon. Your olfactory hairs are cilia that project from the receptor cell dendrite. The nasal hairs that we normally see, those are vibrissae. Those are not your olfactory hairs. So these olfactory hairs are much, much higher up in your nasal cavity and much smaller. Excuse me. Because remember, these are housing chemoreceptors for a specific odorant. So these are going to be very, very small hairs that will perceive smells depending on which cells are stimulated. So depending on which of these cells on the olfactory hairs are stimulated will determine which pattern of nerve signals get sent to the brain to interpret smells. Your olfactory nerves, your cranial nerve 1, are bundles of olfactory cell axons that project through the skull's cribriform plate and enter the olfactory bulbs. So you have your olfactory bulbs, your cribriform plate, and then these projections of the olfactory nerves going into the cribriform plate, extending into the olfactory receptor cells. You have a pair of olfactory bulbs. They're ends of the olfactory tracts located under the brain's frontal lobes. So frontal lobes of the cerebrum, underneath those, inferior to those, are these olfactory bulbs. You have one on each side. The olfactory nerve fibers synapse here with the mitral cells and tufted cells, and their connections form the olfactory glomeruli, which is structural. Your olfactory tracts, again, you have pairs of them. They're axon bundles of mitral and tufted cells on the inferior frontal lobe surface. So inferior frontal lobe surface, still in the same place. And they project directly to the primary olfactory cortex in the temporal lobe, the hypothalamus, the amygdala, and other regions, not through the thalamus like other sensory pathways. Remember, it's going to all these different places because smell is triggering a lot of different things. It's talking about fear. It's talking about memory. So smell brings up a lot of nervous structures for us. There's a lot of CNS structures. And so these are going directly, these olfactory tracts extend directly into all these different areas. Okay, detecting smells. We have to sniff repeatedly or breathe deeply. If you're just breathing normally, unless a smell is really intense and it's right there in front of you, you're not going to smell it. Usually when you have to go smell something, you take a big whiff of it. That's why like when we do smelling things like a chemistry class, you're supposed to waft it towards you. So as opposed to you bringing air in, you kind of gently push air in towards you. So that way it has much less risk of burning and harming that olfactory epithelium. The mucus that's secreted contains odorant binding proteins. that help to bind those odorant molecules. The olfactory sensations begin when odorant binds to protein and protein stimulates the receptor cells. These are rapidly adapting receptors. Your book gives that excellent example about using the trash can, how you can quickly, as intense of a smell that is, you can actually adapt to it pretty quickly when you're just near it. It's an indirect pathway, so you have a G protein and receptor cell activates the adenylate cyclase to convert to cyclic AMP. Cyclic AMP leads to opening of ion channels for sodium and calcium, causing depolarization, and that action pendulum is triggered on the axon and is conducted to the glomerulus. Then you have secondary neurons conducting signals to various central nervous system areas, the cortex, the hypothalamus, and the amygdala. So this is just how that nerve signal is transported through or transduced through the central nervous system. You have olfactory sensations binding to those olfactory receptors, odorant molecules binding to those olfactory receptors. That's going to open up activated G protein cascade that will cause depolarization through opening sodium and calcium channels, and that will then be conducted to the glomerulus, which is then taken to the central nervous system structures, the brain structures. Right, gustation, your sense of taste. Detection of taste notes, right? So if olfaction was the detection of odorants, gustation is the detection of taste notes. Gustatory cells are chemoreceptors within the taste buds. So again, chemoreceptors, we're detecting molecules here. Our tongue has different papillae that are going to house different taste and structures, gustatory structures. You have the filiform papillae, which are short and spiked. They have no taste buds. They're there just to help manipulate food. On the anterior 2/3 of the tongue surface. Your fungiform papillae are mushroom-shaped. They contain a few taste buds and on the tip and sides of the tongue. The foliate papillae are not very well developed in adults. They're usually only in early childhood, housing a few taste buds and on the side, the posterior lateral tongue. And your valate papillae are the largest, the least numerous, but they contain the most of the taste buds. You have a row of 10 to 12 along the posterior dorsal tongue surface, and within each of these papillae are going to be taste buds lining them. So here is the root of the tongue. These are the valate papillae that are going to be housing the majority of our taste buds. Your taste buds are onion-shaped organs housing your taste receptors. Just like with the olfactory receptors, there were 3 cells. Our taste receptors have 3 cells. Our taste buds rather have 3 cells. Your gustatory cells detecting taste notes. Notice this, they only live 7 to 9 days. Your oral cavity is that epithelium. All the cells in your oral cavity are quickly replaced just because of the high abrasive environment that it is. So gustatory cells, your taste receptors are replaced. Taste cells are replaced frequently. You have supporting cells, sustaining gustatory cells, and basal cells, which are stem cells replacing gustatory cells. So very, very similar to olfactory cells. Okay, gustatory cells are neuroepithelial chemoreceptive cells of taste buds. A lot of big words, it's okay. Neuroepithelium, all right. We know neuro for nervous system, epithelial for like the epithelium. Chemoreceptive, so these are going to be chemical receptive cells. You have gustatory microvillus is taste hairs that forms dendritic endings. So just we have olfactory hairs, we have taste hairs, which may kind of gross you out that there are taste hairs on your tongue. Your microvillus often extends through the taste pore to the tongue surface so that it can be stimulated by these taste notes, these tasty molecules, which dissolve in saliva and stimulate your microvillus. So just like we needed mucus to stimulate the olfactory cells, we need saliva to help dissolve and stimulate the microvillus. All right, the gustatory pathways. What is the nervous system pathway for gustation? You have sensory neurons connected to multiple gustatory cells in the tongue that project to the medulla, so going down into the center of the tongue. And the anterior parts of the tongue, the sensory neurons are part of the facial nerve. Posterior is the glossopharyngeal nerve. So 2 different cranial nerves have sensory neurons in the tongue. The secondary medullary neurons project to the thalamus. So you have these first ones going from the gustatory cells to the medulla of the tongue, where they then synapse with different parts of with different cranial nerves. Your secondary medullary neurons go then to the thalamus. The tertiary thalamic neurons project to the primary gustatory cortex and the insula, so deep to the temporal lobe. So you're going from your tongue to your secondary cranial nerves into your actual cerebral lobe. There are 5 basic taste sensations spread over the tongue. There used to be, and I definitely remember doing this in school, dropping different things like lemon juice or sugar water onto your tongue and trying to determine what the taste map is, but that's not a thing. I think actually the next slide does that, but I just want to go and get out there. The taste map on your tongue is not a thing. You have taste buds, they detect all range of things. You have 5 different tastes, though, that we can generally detect: sweet, salt, sour, bitter, and umami. Sweet is produced by organic compounds, sugar, artificial sweeteners. Salt is metal ions, sodium, potassium. Sour is from acids, things like vinegar. Bitter is from alkaloids, so unsweetened chocolate. And umami is related to amino acids. Bitter is one of the tastes that Westerners, especially particularly Americans, we get the least amount of. The bitter taste is one that we tend to kind of crave the least, and that we eat the least. If you think they have unsweetened chocolate, but also things like really dark leafy greens, those are all bitter. And a lot of times there are studies that have shown that hunger, when you eat a meal and you're still hungry, it's partly because we need this kind of bitter component. If you add some kind of leafier green, something that has just a little bit more bitterness, don't add unsweetened chocolate and just eat that as your part of your meal to try and round it out. But if you can add a little bit of bitterness to your meal, it does kind of help you feel more satiated. You kind of have hit all of those flavors. Okay, so transduction and gustatory cells, so converting these chemical signals to electrical ones. For sweet, bitter, and umami taste, the tastants are molecules. So your tastants are binding to a specific cell membrane receptor. The G protein is activated, causing a second messenger formation, just like we did where we formed cyclic AMP in the olfaction. And then you get your cell depolarized. So sweet, bitter, and umami, they're going to be molecules, activated G protein to depolarize your neural cells. Salt and sour tastants are ions, so they depolarize the cell directly. So either molecules going to be indirect or ions binding directly. Depolarized gustatory cells release neurotransmitters stimulating primary neurons either in the cranial nerve 7 or 9, your facial or your glossopharyngeal. Your primary neuron in the cranial nerve brings a signal to the nucleus solitarius within the medulla. Your medullary activity triggers salivation and stomach secretions, so kind of prepping ourselves for digestion. Nauseating stimuli instead trigger gag or vomiting responses, reflexes. The signal then from the medulla is relayed to the thalamus, and then it's relayed to the primary gustatory cortex for conscious taste. Taste is integrated with temperature, texture, and especially smell. And noted that food has less taste if olfaction is blocked. That is an actual thing. If you can't smell something, you're not going to be able to taste it as intensely, which is also why if you have to eat something that kind of smells gross, then you blocking your, kind of holding your nose and eating it does actually help cut down on the taste. Right, and this is just kind of showing that pathway, that gustatory pathway. So starting with these odorant molecules are going to be going on their olfactory pathway, but then you can see these neurons in the medullary part of the tongue going up through the medulla, the thalamus, and to the gustatory cortex. Okay, now we're getting into structures of the eye for vision. We're going to get through all of the anatomical structures of the eye, and then the second part of this chapter 16 will be on actual vision and hearing equilibrium. All right, accessory structures of the eye are attached to or around the eye. Sorry, my dog is chewing on things. They include the six extrinsic eye muscles: your eyebrows, your eyelids, eyelashes, conjunctiva, and lacrimal glands. Your eyebrows are located along your supraorbital ridge, so above your eye, supraorbital ridge. They aid in nonverbal communication, surprise, and prevent sweat from dripping into your eyes. Your eyelashes extend from the margins of the eyelids. They prevent objects coming into contact with the eye and can initiate your blink reflex. Your eyelids are palpebrae. They're made of a fibrous core, the orbicularis oculi muscle, and very thin skin. The upper eyelid is raised by the levator palpebrae superioris muscle. So upper eyelid, palpebrae superioris, raised levator. So muscle name tells you exactly what it does and where it is. And your space and sweat glands are at your eyelid base. When they are infected, you get a sty. You have conjunctiva, which is the transparent lining of the eye and eyelid surfaces. It's specialized stratified columnar epithelium, so it's going to be fairly thick epithelium, not going to be really easy to get through. It contains numerous goblet cells to moisten the eye, many blood vessels to nourish sclera, and abundant nerve endings. So goblet cells moistening, blood vessels nourishing. It does not cover the cornea, so it doesn't interfere with any light passage. And when it becomes infected, you get your pink eye, your conjunctivitis. Just breaking this down in case you did not know where your eyebrows are. Have you ever noticed, one of my favorite things is to figure out which celebrities don't actually have eyebrows and looks very normal. Whoopi Goldberg is that kind of famous one that, she doesn't have eyebrows. It's very interesting like to see, it's like, oh wow, some people you never notice they never had eyebrows. Your eyebrows, your eyelids, you have your pupil, which is the dark center where the light passes through, the iris being the colored portion of the eye. You have your sclera, which is this whitish portion covered by that ocular conjunctiva. Remember that transparent lining over top of it is the conjunctiva. The palpebral fissure is the space between your eyelids. It can be wide or it can be almost non-existent, just depends. You have your lacrimal caruncle, so part of the lacrimal gland. And I think that's good. All right, there's just, this is just kind of more external anatomy of the eye structures. This palpebral fissure, which between the eyelids, you have your lens. You have your lens here. You have your pupil right in here. Your iris is going to be this colored portion around it on this side. Your cornea covering all of it. Okay, lacrimal apparatus. It produces, collects, and drains fluid. Lacrimal fluid itself is mainly water, sodium, antibodies, and lysozymes, which is an antibacterial enzyme. Lacrimal fluid works to lubricate, cleanse, and moisten the eye. It reduces eyelid friction, so the palpebrae can blink easily. Defends against microbes, and oxygenates and nourishes the cornea. Your lacrimal gland produces fluid and secretes it through the ducts, a duct system, located in the superolateral orbit. And our blink reflex. Blinks wash fluid over the eye about 15-20 times per minute. Here is that lacrimal apparatus. So you have this lacrimal gland up in here. It starts to secrete its fluid that washes over the eye and then drains in through these lacrimal ducts, the sac, and then out to the nasal cavity. Either out through the nose or back through the throat. All right, so the eye is almost spherical. It contains two cavities, the posterior cavity behind the lens that has the permanent vitreous humor, and the anterior cavity in front of the lens, which contains the aqueous humor. So anterior aqueous humor, posterior vitreous, you have to remember that. Posterior permanent, there you go. The wall of the eye is formed by 3 tunics. Tunics are just layers. So you have your fibrous, your vascular, and your retina tunics. External, middle, inner. Right, there's just a view of that. I'm not sure why they include this picture, but you can see kind of the visual axis, and then just off from that is this optic nerve. Okay, so you have your outer fibrous tunic, which is both your sclera and your cornea. The sclera kind of covering around the kind of the eye that's in the orbit that we can't see, the cornea being that front portion. You have your vascular tunic, which is your iris, your ciliary body, and your choroid, so vascularized, so it's going to house your blood vessels. And then your retina, which is your internal layer, and it has 2 layers. It has this neural layer and a pigmented layer. We'll get into both of those. Here is just a further breakdown of these eye structures. This is really good to go back and refer to as we go through this if you're having trouble kind of keeping what things are straight and where kind of light is passing through. This is a great picture to go back to. Okay, the fibrous tunic is the tough outer layer. It's composed of the sclera and the cornea. Your sclera is the white of your eye. It has dense irregular connective tissue to provide the eye's shape. It protects the internal components, and it's an attachment site for extrinsic eye muscles. So again, it's very much a connective tissue. It's got protecting internal components, providing shape and structure, and an attachment site. Your cornea is the anterior convex transparent window, so anterior convex transparent window across the front of the eye. It has an inner layer of simple squamous epithelium, a middle layer of collagen, and an outer layer of stratified squamous epithelium. Collectively, this is all called your corneal epithelium. I'm not going to ask you to know that specifically, the components of the corneal epithelium. It contains no blood vessels, remember to not block with the passage of light, and it is going to refract light. We'll look in a minute at what refracting light is, or actually I think in the next lecture we'll do refracting light, but it's just kind of bending light. Your vascular tunic is the middle layer with many blood vessels, lymph vessels, and intrinsic muscles. It has three parts: the choroid, the ciliary body, and the iris. The choroid is very extensive. It's in the posterior region of the vascular tunic. It has many capillaries to nourish the retina and melanocytes to make melanin to absorb extraneous light to help prevent light scattering. The ciliary bodies contain ciliary muscles and processes. They're just anterior to the choroid. Your ciliary muscles are bands of smooth muscles connected to the lens. These, when the muscles contract, loosen suspensory ligaments, altering your lens shape. So we'll get into accommodation and how the lens shape changes to adjust to seeing things closely or seeing things closely or seeing things farther away. We'll talk about that in a little bit. But it's the ciliary muscles that are partially responsible. And your ciliary processes contain capillaries secreting that aqueous humor in the anterior chamber. Also part of your vascular tunic, your iris. It gives the eye color. It's the most anterior region of the uvea. It contains smooth muscles, melanocytes, vessels, and neural structures. The iris also divides the anterior segment into the anterior chamber between your cornea and your iris, so that outer transparent structure and the colored structure, and the posterior chamber between the iris and the lens. So you have anterior between that and the posterior there. Anterior chamber and posterior chamber are both part of the anterior cavity, though. So you have your anterior cavity and your posterior cavity, and then your anterior cavity has both an anterior chamber and a posterior chamber. I did not decide that. That's not my fault. The pupil is the opening in the center of the iris connecting these two chambers, and the iris controls your pupil diameter. So you have sphincter pupillae muscles and dilator pupillae muscles. So pupillae muscles tell you that they're going to be acting on the pupil. Sphincter muscles and dilator muscles. When sphincter muscles, a sphincter is a constriction, so when it contracts, it's going to close things smaller. When dilator muscles, when they contract, they're going to dilate. They're going to make openings larger. So they're both contracting, but they're contracting in different ways because of the muscle fiber directions. So sphincter pupillae are concentrically circular fibers. That seems redundant. Concentrically circular fibers that constrict pupil with parasympathetic activity. So that is something important to remember. Sphincter pupillae are innervated by parasympathetic nerves, specifically cranial nerve 3. Your dilator pupillae muscles are radially organized, and they dilate pupil with sympathetic nervous system activity. So the reason that our dilator muscles are innervated by sympathetic muscle activity is because when our sympathetic nervous system is when our sympathetic nervous system is active, we're trying to take in more light. We need to get in more information because that's typically our fight or flight response. And so by dilating the pupil, we can then take in more light and receive more stimuli. And our parasympathetic system, when we are resting and digesting, we don't need to be getting as much stimuli. The pupils can constrict, and we don't need to get as much information. We're not in an emergency situation. The pupillary reflex alters the pupil size in response to light, so that is independent. The light-dark response is independent to the parasympathetic-sympathetic activity. Increased brightness leads to constriction. Decreased light leads to dilation. So pupillary constriction under bright light, the sphincter pupillae contracts. Pupillary dilation in low light, the dilator pupillae contracts. So your retina is the internal or neural tunic, the internal or neural layer of your eye. It has both the pigmented and the neural layers. The pigmented layer attached to the choroid, and it provides vitamin A to photoreceptors, and it absorbs stray light to prevent light scatter, similar to melanocytes. The neural layer houses the photoreceptors, your rods and your cones, and the associated neurons. and it receives light and converts it to nerve signals. So it's going to be receiving, it's going to be part of transduction, right? So it's going to be receiving the stimuli and then converting it to an electrical stimulus, an electrical signal. Also, you have the ora serrata. Serrata meaning like serrated, so it's a jagged edge. It's the boundary between the photosensitive and non-photosensitive parts of the retina, the neural and pigmented layers. Non-photosensitive part is the, I'm sorry, no, that's not correct. The boundary between the photosensitive and non-photosensitive. The non-photosensitive is part is anterior. It covers the ciliary body and the posterior side of the iris. All right, so the neural cells form 3 sublayers. You have photoreceptor cell layer, the bipolar cell layer, and the ganglion cell layer. Again, those names should go ahead and tell you something about what they're going to be doing, what their function is. So the photoreceptor cell layer is the outermost neural layer, and it contains your rods and your cones. It contains your photoreceptors. These contain pigments that react to light. The bipolar cell layer, their dendrites receive synaptic input from the rods and cones, so they're going to be receiving input from the photoreceptive cell layer and then transmitting that to the ganglion cell layer. Ganglion cell layer is the innermost neural layer. Their axons gather at the optic disc and form the optic nerve, and they're capable of inducing action potentials. Okay. Yes. I just also want to kind of make a point. When it says innermost neural layer, it's referring to here. So when it says innermost, it means innermost in relation to deepest, sorry, I'm touching my screen like you can see me touch my screen, deepest into the skull. So this portion right here, you know, we're talking about our retina, we're talking about our neural retina. This kind of more superficial layer, that's going to be where your photoreceptor cell are. As you go deeper into the eye structure, you're getting more towards your innermost. So innermost doesn't mean innermost as far as medulla of the eye. It means innermost as in closest to the skull. So just, you know, if that confuses you more, then just ignore me and, you know, don't think about it. But okay, here we go. This might help us. So as we go in, we start with this pigmented layer, we get into our neural layer, and as we go deeper and deeper in towards the skull, towards the back of the head and the brain, that's when we go from the photoreceptor cells to the bipolar cells to these ganglionic cells. And then here we have the cells of the optic nerve. Okay, your more components of the retina. Your optic disc contains no photoreceptors. It is your blind spot. This is where the ganglions exit towards the brain. So this is the part where your optic nerve is going to be exiting. Your macula lutea is the rounded yellowish region lateral to the optic disc, so just to the side of the optic disc. It contains the fovea centralis, which is your central pit. It has the highest proportion of cones, very hardly any rods, and the area of sharpest vision. We'll get into the actual functions of rods and cones in a little bit. So the fovea centralis, area of sharpest vision. You have your peripheral retina, which contains primarily rods and functions most effectively in lower light. Rods are best in lower light. Cones work better in brighter light and with color. There's just an internal view of that, so you can see the optic disc over here. It's kind of that convergence of the optic nerve. You have your macula lutea with the fovea centralis right in the middle of it. Okay, so the lens. The lens will change shape to focus light on the retina into the appropriate portions of the retina. The cells within it have lost organelles and are filled with a crystalline protein. And the lens itself is enclosed by a dense fibrous elastic capsule. The shape of the lens determines the degree of light refraction, so the degree that light will be bent. And the shape is determined by the ciliary muscles and suspensory ligaments that hold the eye, that hold the lens. When viewing the objects 20 or more feet away, so objects at a greater distance, the muscles relax, the suspensory ligaments are tensed, and the lens flattens. So if you're trying to see something that's further away, the lens of your eye will flatten. Whereas if you're viewing an object that's closer to 20 feet, the lens does what's called accommodation, where the ciliary muscles are tense, the suspensory ligaments are less tense, and the lens itself kind of becomes more spherical to bend light more. So here you can see lens shape for distant visions. The lens gets more flattened, whereas lens shape for near vision, this accommodation, the lens really gets this more spherical shape. Okay, your vitreous and aqueous humor. Your vitreous humor in the posterior cavity It's a transparent gelatinous fluid behind the lens. It's permanent fluid, first produced in embryonic development. So during this first 8 weeks of development, we produce this vitreous humor. And it helps to maintain our eye shape by kind of keeping the pressure within the eye. So it supports the retina, keeping it flush against the back of the eye. Right? So it's just fluid that helps to kind of fill things out. Aqueous humor is the transparent watery fluid in the anterior cavity in front of the lens. It is continuously produced by the ciliary processes and helps to nourish and oxygenate the lens and the inner cornea because there aren't any vessels in here. So it has this aqueous humor that's doing that for it. Okay, and actually that's where we're going to stop for this one.